Okay, are you ready? Okay, I'm ready too. So we start with the sponsor talk, and then we will have a lightning talk, and then we will have the the GRG study talk. Okay, and so I'm very excited to that actually N6 is the sponsor for this session because I really thought that that was the coolest tech that I saw at AGBT last February. And so Chris from N6, it's like a K-pop group.
Well, thank you very much. I love coming to ABRF. There was more karaoke at AGBT than here so far, but you guys need to up your game a little bit, and we'll make it a good time. So N6 is a new company. We've all come from different backgrounds within PacBio, Biorad, ABI, Illumina, 10X, that sort of thing. And effectively what we've built is an instrument to address that bit between sample prep and sequencing that is the most very laborious and expensive aspect. And so this is the world's first and only, really, thermocycler with individually controlled wells. And that's how we came up with the term ICON, individually controlled wells. And when I say that, I mean we have 96 miniaturized thermocyclers inside of there. So you have to kind of flip your thinking a little bit from a big aluminum block to treating every sample individually. And we've got our management here, our CEO, and the mission when they designed this was the idea of don't make you change your consumables, don't make you change your assays, take everything you have today, but address the biggest bottlenecks in the overall NGS process, and at the same time give better data. Everybody knows that PCR is required, but it's also not your friend, right? And these days, people just do it by brute force, either by lots of purifications, lots of manual interventions, or robotics. So we tried to go through every step in the process, starting with the individual quant. We say, load whatever you want into the instrument. We don't care about the input. You don't have to spend a bunch of time quantifying the samples going inside. Obviously, we don't do a lot on the adapter ligation, but when you're talking about removing biases from over-amplification, so if you've got two different samples with a little bit or a lot of DNA, some of them will drop out, some of them will over-amplify. We're going to treat those samples as an individual sample and amplify them correctly without introducing artifacts. At the end of the day, you usually purify every single sample, quantify every single sample, and then normalize. We're going to bring all of your samples out of the PCR thermocycler at the exact same level. So that allows you to save a tremendous amount of both spry beads as well as manual qubit, different quantification methods to understand the different amounts of material in each individual sample. But at the end of the day, we also want to make sure that you have the best sequencing quality. So if you over-amplify, you're going to have a lot of PCR duplicates, you're going to have a lot of artifacts, adapter dimers that might poison the well. So all of that comes down to the cost of your experiment. And the goal of this is to go back to the beginning, amplify every sample correctly from the first place. Don't treat them the same. So, how do we do this? Basically, when you see this amplification curve, what we tell you to do, and we've got a number of different normalization methods, and if you come down to our booth 208, we'll explain the different methods, but you can tell the instrument to amplify every single well to the exact same level. So you set that threshold, and every single reaction is going to then amplify to that level and then shut off. And I'll show some data a little bit later, but we have a number of different methods. You could amplify to maximum yield. You could amplify to the green line, which is limit your PCR amplification as much as possible. We have different methods that actually do that, but at the end of the day, we're treating every single sample individually. And we do that by basically miniaturizing the thermocycler into 96 individual thermocyclers within the block. So it's no longer one big hunk of aluminum, it's individual thermocyclers. Each one of those little thermocyclers has a thermistor on it, so it measures the temperature in real time. So if there's any crosstalk from any well next to it, it knows it at 2 milliseconds, and it adjusts the temperature, so we have precise control. And again, the goal, the non-negotiable aspects of the design of this instrument is use your standard plastics. Work within the range of 10 microliters to 100 microliters for those of you doing those really funky single cell assays. We're basically going to add cybergreen to your reaction to measure it. That's been around for a decade. Everybody knows that doesn't affect the quality of your data. And we're going to auto-normalize your libraries. Now, if you're doing NGS libraries, we can auto-normalize that. But if you're actually optimizing different assays and developing your own reagents, then you can actually have a lot of different flexibility. You could set up 96 different reactions on that 96-well block to do your DOE in one run instead of 2 months worth of experimentation, including setting, and this is what we actually recommend for our quote-unquote optimization. We recommend that you take your samples and do fixed cycles through 2, 4, 6, 8, 10, 12, all the way through, identify the optimum number of cycles, and you're set for the rest of your life with that particular assay of the optimum amount of optimization. And we even have a zones mode. So if you want to run 3 different PCR profiles at the same time, or 96 different profiles, you can set that up on the block and run it all parallel to each other. So I think the crowd here is mostly interested in NGS for the workflow and cost savings. But if you're optimizing technology or developing assays, it actually is very helpful to develop your technology, develop your assay in parallel. All right, here's an example of the auto-normalization. On the left-hand side, we set the threshold. On the right-hand side, you see at the bottom of the panel the temperature profiles of every individual well. They're going up and down as they cycle through. The first sample, the green one, hits the threshold, and that well in the bottom panel stops. It goes to a cold degree hold, and the rest of the wells continue to amplify. And so now all of those subsequent wells will keep amplifying till they hit that same level, and then they're able to be taken out of that individual reaction. And you take all of these steps in the pink and take all of your unpurified libraries, take one aliquot from each one of them, do one spry bead cleanup. One spry bead cleanup of the entire plate, and now you have normalized libraries. So when you think about the cost of spry, the cost of all of that labor, being able to get that in one example. So this is a Na12878. We start out about 20 nanograms, and we did a dilution series all the way down to about 0.5 nanogram. And as you expect when you do traditional PCR, the higher inputs give much higher yield, all the way down to the lower inputs at about 0.5 nanogram. When we actually do the auto-normalization, we amplify those low inputs to the exact same level. So we took 1 microliter of every single one of those, every single one of the bottom reactions, pulled them together, and then sequenced them. So you can actually, that's read counts. This is sequencing data right here. And you can see one of those reactions just stochastically needed one more cycle, because we can't do a half a cycle, right? One more cycle to hit that threshold. But we tell you that. So you could say, oh, I should have taken 0.5 microliter instead of this one, but we wanted to actually show that. So this gives you a very consistent result of auto-normalized libraries. But it doesn't just impact workflow. It also matters about data quality. So these are FFPE samples that we got from a collaborator. They range from a DIN, so a quality score of 1, all the way up to a quality score of 8. And we amplified these libraries both with standard PCR and our PCR. And the goal of this researcher, this collaborator, was to put as much DNA as possible, because they wanted to do exome sequencing, so diversity was really, really important to them. And you could see that After about a DIN of 3, the yield really starts to drop off because you have these jackpot reactions of your adapter dimer. You have overamplification of non-productive product. We were able to stop the reaction before that happened and actually rescue low quality samples. So if you're doing single cell and you've got a bunch of dead or dying cells, or mixed 8,000 cells in this reaction, 1,000 cells in this reaction, we can actually get you that, we can actually rescue a lot of those samples. Next is 16S, and basically this is a PacBio run with a 1,500 base pair V9 amplicon of a bunch of soil samples that were sent to us blind. Lots of variability of the input. We ran the collaborators protocol at the top, 25 cycles, and you see that they have typical 16S material. Now, this particular researcher used to put their samples into a real-time system, and they used to monitor it, like sit there at the instrument, and whenever one well hit the level of amplification they wanted, they would open the instrument up, they would excise that one well, they would pull it out of the thermocycler, because they knew that the true diversity was what they saw right here. So you can actually maintain very high quality data with the known effects of PCR duplication. So we've got all of this data down at our booth. Come see it, come see the instrument. But overall, it's a mix between improvement of overall workflow and overall data quality. We all know and love 10X. They have in their protocol the steps to preamp their material, take an aliquot out, which technically reduces your diversity, measure it to make sure you've got enough, re-amplify it if not, and then go on with your library prep. That can all be solved with one ICON PCR. So I'm not responsible for you going off protocol with 10X at this point in time, but it will save you a lot of time and heartache. Beyond that, if you are an assay developer, this is an instrument that will give you a DOE type of experiment in a single run. We can set a gradient, a temperature gradient, or different profiles on the individual block, but at this point in time, we're really specifically setting temperatures. If you think about the traditional old blocks, they actually have kind of not truly a gradient from the top up to the bottom. We can tell you exactly what temperature it is. We can do rows, we can do columns. So those of you who are trying to optimize applications, this is the technology to do it instead of having either 20 different thermocyclers lined up and running them parallel, or monopolizing a thermocycler for a long point of time. So at the end of the day, we are, this is still a thermocycler, right? It's using the exact same profiles, it's using the exact same reagents, we're just giving you control at the individual level. So this is not shocking results. These types of data showing PCR biases have been around for a long point in time. We're just now giving you the control to actually run that, and we're going to save you a lot of money in the operation of your high throughput genomics, as well as rescuing samples on the small end. So with that, I will close. Thank you so much, because the talk a little bit later today talks about the impact of of that kind of research. And if you've got any questions, please come down to 208. We would love to talk to you about the technology.
Wait, I have a question.
Oh, you got questions?
Don't leave yet. So I think there is one important question. Is it available now to purchase?
Very good question. Thank you. So we are in early
Are you
access.
shipping to Switzerland?
For you, anytime. So we are in early access right now within the Bay Area, and later on this summer we're going to be shipping to all of the other places. So any other questions while we're at it? All right, thank you all.
Thank you.
Thank you so much.
For the lightning talk, we have Amy Leindacker, and she will talk about high throughput chipset.
We specialize in an ultra-high resolution version of ChIP-seq called ChIP-exo. We start with cross-linked cells, fragment the chromatin, and then immunoprecipitate with an antibody specific to the target protein. Then we use lambda X nuclease to digest the DNA up to that protein cross-link site, prepare libraries, and sequence. The X nuclease step makes this a stranded assay, such that the 5' end of the top strand and the bottom strand mark the protein cross-link site. These define the footprint of the protein on the DNA, genome-wide at base pair resolution. So I'm showing on the bottom the high resolution of ChIP-exo in that red peak compared to traditional ChIP-seq in that green wide peak. On the right is the example of ChIP-exo in human cells for the transcription factor USF1, which is known to bind to this motif CAC-GTG. And I've plotted the ChIP-exo reads centered at the USF1 motif, plus or minus 250 bases on either side. And you can see these really sharp peaks. The top strand is plotted on the top, the bottom strand is on the bottom, and you can see the footprint of USF1 across those sites. We've now automated our ChIP-exo for high throughput on the Eppendorf EpiMotion 5075T. And we've paired that with our Element Biosciences AVITI sequencer through our custom LIMS system, the Platform for Epigenomic Research, or PEGR. To test our automated workflow, we ran a chip validation screen using antibodies from the NIH Protein Capture Reagents program, and we chose 85 antibodies that had failed previous validation in our previous study. Of the 85 antibodies tested, 17 pulled out clear DNA binding motifs, including P73, which pulled out the P73 DNA binding motif, and SMARCC1, which pulled out the NRF1 binding motif. Now, because cross-linking freezes protein DNA complexes in place, we can also look at DNA-bound protein complexes. So MAFG and BAC1 both pulled out the MAFG DNA binding motif, and this makes sense because BAC1 and MAFG make a heterodimer. If we plot those ChIP-exo reads, you can see not only MAFG at the MAFG motif, but also BAC1 and additional proteins, including STAT3. We can look at larger complexes also. Here I'm showing at the GATA3 motif an array of transcription factors that form complexes, and we can even see subcomplexes within this region down to where their location is, +16, +28. And to further characterize the composition of these complexes, we are planning experiments with the Cornell proteomics core, pairing ChIP with mass spec to further characterize these. Thank you very much.
You can ask a question now or later, after the session. If you have a pressing question for her. Okay. Now the moment of truth. Just have to change computers. Yes, maybe. And your WhatsApp.
Yeah.
So I will give the intro, and then Natalia will. So my name is Catherine. Yay. And I will give the introduction, and then the data, and then after the introduction, then it will be Natalia Zajac who will present the rest of it, the important stuff, and then also the rest of the GRG is also here to, if you have questions, to answer the question, to answer questions. So please listen very well and have questions later. Okay? Promise? Okay. Yeah, so today we will present the GRG study on the impact of PCR duplication on RNA-seq data generated using the NovaSeq 6000, NovaSeq X, AVITI, and G4 sequencers. And so this is a study of the genomics research group. And so these are the current members of the genomics research group. And if you want to join us, please send me an email because we're always looking for new members. And yeah, so maybe after you listen to our study, then you can, there will be a follow-up for the next year. And then, yeah, so if you're interested in these kinds of studies, please join us. And special thanks, of course, to Lutz because he helped us with sequencing on the AVITI. So, so first, I want to just the intro, just to explain what duplicates in NGS data is. So duplication rate is we define it as any two reads that share or any reads that share the same 5' and 3' coordinates. So in this study, we define duplicates as the same fragment sharing or having the same UMI. And so the duplicates are produced mainly during the PCR step in the library prep step of sequencing. And so many analysis softwares remove the PCR duplicates, but unfortunately, but it's an accepted fact that there are duplicates. And so for example, for RNA-seq data, sometimes we see like 40%, 60%. The problem with that is that it eats up reads, right? So because you don't really need duplicates, it's not useful. So so then the read, so you sequence a lot, which is good for the sequencing companies, but then yeah, but then so you can just throw it away, but then it's more expensive, right? So during the library prep workflow, what steps produce the duplicates? So basically, you can produce duplicates during the PCR. Yeah, sure, that's the point. And then during the clustering step, and then also during the sequencing step. So these are the different kinds of duplicates. So PCR duplicates, sister duplicates, clustering duplicates, optical duplicates. And if you have been sequencing for a while, so I say this ancient slide that explains what duplicates are. So for example, an optical duplicate is defined by a single cluster that's called 2. So that's called an optical duplicate. If you want to, what is a clustering duplicate is, so the clusters during cluster generation, two nearby clusters are, adjacent clusters is basically, during cluster generation, a library occupies two adjacent wells. So this is mainly for pattern flow cells. And then there are the sister duplicates where the complementary strands produce or treated are basically, called the 2. Yeah. And then there are the PCR duplicates, which is, the actual PCR. So this is what we are looking at, the PCR duplicates. Long story short. So this study, we started basically just wanting to look at duplicates. So it was we started very simple. So we just wanted to see what is more important, the input amount or the PCR cycles. That's the point. I mean, that's just what we wanted to do. And then, so when we got the data, the new sequencers also started popping up, And then we bravely decided, I mean, we had to, shall we do it? Shall we not do it? We were a bit afraid because, if the results are not good, then the sequencing companies won't love us anymore. So, so we decided that, okay, shall we do it or shall we not do it? Shall we do a comparison? But we have to do it as not biased as possible, of course, because we want to be loved still. Yeah, so what we did was, it's always the question in everybody's minds, right? So when you do this conversion to use the new sequencers, does it change the library complexity? Does it produce more duplicates? So what's going on, right? And then at this point, we already had like the perfect sample for it because we have the libraries with the PCR duplicates, and then the UMI. So we could actually figure that out, and it's just a matter of sequencing the different libraries with the new sequencers. So we decided that we will be brave and do it. If anybody gets angry with the result, it's Chris Mason's idea, okay?
He's the only one not here.
and I can say that because he's not here. Okay. Yeah, so that's the goal of the experiment, the goals, the objectives of the study. And so for the material, so we used total RNA total RNA from Thermo, and then we use actually the NEB kit because they have adapters with UMIs already, so we don't have to make our own. And also the NEB Ultra 2 live RNA has a very wide input amount. So for experimental setup, we did a serial dilution starting from 1 microgram to nothing, to 0 nanogram. And then we used adapters with UDIs and UMIs. And then we chose 3 PCR cycles per input amount. And then the libraries we pulled together and then sent to these different centers for sequencing. Because so at this point, it was like last year, there was not so many installs of the new sequencers, so we could only send it to very specific places. Yeah, and then we only actually had the 6,000 data. We did it ourselves in Switzerland. And then when we got the NovaSeq X, then we did it also then because we still have the libraries. Yeah, so we had the serial dilution, and then we did the libraries, and then so we classified the PCR cycles into low, mid, and high, for the input amount. And then we divided it for the different sequencers. And then for the G4, we sent it out to Ioannis. And then, so basically it's just like you just PCR in the G4 specific adapters. And then for the AVITI, you put this splint oligo, and then, and then you make this circularized library. And then Natalia will tell you all about the results.
Hi everyone, so my name is Natalia. I also work for the Functional Genomics Center Zurich. I'm the bioinformatician there, and I'm here to present the results of the study. So to explain the bioinformatics workflow, what we did after sequencing, we performed quality control of the data, and that included the Fred quality scores, the contamination, and so on. We then subsampled the reads to 2 million reads per sample, because not all of the samples got above 2 million reads. And we aligned it to a human genome. We deduplicated the reads, the samples using UMI tools, using the UMI and the coordinates, the mapping coordinates, and then we assigned fractional reads to counts, fractional counts of reads to genes, and we profiled the gene expression of each sample. The aim, so to recap the aims that Kathy already mentioned, we wanted to investigate the impact of the input amount on the rate of PCR duplicates and data quality, the impact of PCR cycles, which we chose the 3 different PCR cycles, on the PCR duplicates and data quality, to evaluate the efficiency of the library conversion on production of the rate of PCR duplicates, because the conversion adds several PCR cycles, so we expected that that might add additional PCR duplicates to the data and lower data quality, so we wanted to evaluate that. And then finally compare the data quality between the different sequencers. We wanted to evaluate, see how the PCR duplicates impact data quality, and how that impacts the results. And the results included the mapping, the number of detected genes, and the gene expression profile. So to start with the raw data quality, the FRET quality scores between the samples were very comparable. AVITI did have a higher average of 43, but the FRET quality score ranged between 36 and 43. We evaluated the sequencing error rate by using the number of mismatches in the mapped raw reads. We observed a decrease in the sequencing error rate with the increase of the input amount, but also for input amounts between 4 and 61 nanograms, we observed an increase in the mismatch rate or the sequencing error rate with the increase of the PCR duplicates. So as you can see, there are several of these higher peaks here, and those are this is the high PCR cycle category. So this had an increase in the number of PCR on the sequencing error rate. And you can also see an elevated error rate in G4. Here, you can see a graph of a profile of each sample and the proportion of reads that belong to each of the 6 categories. So this is divided by the sequencers, going from AVITI, G4, NovaSeq 6000, and NovaSeq X. Don't know if you can see that at the back, I'm sorry about that. And the input amounts are organized from the negative control, so 0 input amount to 1,000 nanograms. And the PCR duplicates, so the number of PCR cycles used for amplification go from low to high. And so what you can see from here, that the reads are categorized into 6 different categories. The red indicates the unique reads that were mapped and counted into the gene count matrix. The yellow ones are the unique reads that were mapped but not counted because, for example, as displayed here on this diagram, they mapped to intronic reads. And then the PCR duplicates in green. And then there are 3 categories of reads of the unmapped reads, the low quality reads, and the reads below 18 base pairs. So those we would consider adaptive primer dimers. And you can see that in the Illumina data, without cleanup, there is an elevated rate of so first of all, maybe to address the PCR duplicates, with increasing input amount, you get a decreased amount of PCR duplicates, and with increasing rate of PCR cycles, you also get an increased rate of PCR duplicates. You can see that especially for the low input amounts, the more the sample is amplified, the higher the rate of PCR duplicates. And the green disappears when the input amounts go into high, above 125 nanograms. The rate of primer dimers is high in Illumina data without cleanup. However, in the library conversion decreases these, so there is an additional step of library cleanup, and therefore the primer dimers disappear, but there is an increase in PCR duplicates as we expected, and there is an introduction of contamination. You can see the rate of these blue unmapped reads has increased in negative control, and from those reads, so we estimated the contamination across these samples, and the reads mapping to the human genome increased from 30% to 60%. So there are several steps that you need to be aware of when you process the samples with additional step of library conversion. Now, how does that impact all the 3 different categories of results? So in terms of mapping, you can see here that the removal of PCR duplicates impacts the mapping significantly at low input amounts. The purple indicates the raw reads without any PCR duplicate removal, and the blue indicate the cleaned up data, indicates the cleanup data. And you can see that there is a big decrease for low input amounts up until 125 nanograms, where the removal of PCR duplicates does not impact the mapping, the percentage of the reads mapped anymore. You can also observe that for Illumina data, the rate of PCR duplicates, because it's lower, there is also a smaller difference between the raw data and the cleaned up data in the percentage of reads mapped. And there is a bigger impact of PCR duplicates on the highest category of the PCR cycles. So over-amplification can definitely heavily impact the data. When it comes to the number of detected genes, we do see that the input amount does define how many genes you can detect. There is an increase in the number of detected genes with the increase of input amount, but there is an interaction between the PCR duplicates and the input amount in the number of detected genes. As you can see, between the input amounts of 7 nanograms and 62 nanograms, those amples that were over-amplified have lower number of detected genes. So as you can see here, the comparison is very clear. The higher the number of PCR duplicates, the lower number of detected genes. But there isn't that much of a difference between the intermediate and the low category of PCR cycles. We compared the number of detected genes between the sequencers for the low PCR cycle category for input amounts of 125 nanograms to 1,000 nanograms. And we do detect that 83% of the genes were detected by all sequencers, and 86% of them were detected by at least 3 sequencers. We did investigate the structural differences between these reads that were not detected, and we do not see any bias in the GC content or the width of the genes, so it seems to be a random effect. Could be also an effect of subsampling. In terms of the gene expression profile, we evaluated that using a UMAP, which is a uniform manifold approximation and projection. And it's a technique, for those who are not maybe aware of it, to reduce a many-dimensional data into a 2D space. The dimensions in the data are defined by the number of genes that are measured. And here you can see the samples are colored by the input amount and the sequencer and then the PCR cycle category. You can see that the dark samples cluster in the middle because there doesn't seem to be that much of a difference in the gene expression profile for the samples of high input amount. But the lower the input amount, there is more structure in the data. And so you can see that these little clusters are formed by the sequencers from the same input amount and from the same PCR cycle category. So there is a very heavy effect of PCR duplicates and low input amount on the gene expression profile. We also evaluated that using, we looked at the genes that are expressed, and we selected top 1,000 most variable features according to the gene expression, and we do see that for the highest variability is observed due to the input amount and the PCR cycle category. So here the samples are ordered by the input amount. You can see the top row indicates the color, and the color indicates the input amount. Then there are the PCR cycle categories going from low to high, and then the sequencers. And you can see that there is underexpression detected for those 1,000 top most variable features. So the profile doesn't correspond to those samples from high input amounts, and the structure, for example, for those mid input amounts, the gene expression profile is affected by the PCR duplicates, by the PCR cycle category. Sorry. You can also see that not removing duplicates can lead to an overestimation of gene expression. Here is a comparison of the data of the correlation between the high and the low PCR cycle category, pre-duplication and post-deduplication according to the different input amounts. And you can see that for low input amounts, the counts are extremely high before deduplication, and there isn't much of a difference between low and high PCR cycle category. But after you remove duplicates for input amounts between 7 and 15 or 31 nanograms, there is a discrepancy between high and low PCR cycle category. And the low category has higher counts for the genes detected. There is also, you can probably observe the difference between the colors indicate the different sequencers, and you can observe at 250 and 500 nanograms, there is a difference or discrepancy between the NovaSeq sequencers and the AVITI and G4, and that is due to the adaptive primer dimers. You might have observed that at those input amounts, there are two samples that are affected by primer dimers. Okay, so to conclude, the results are comparable between sequencers, and we see the same association between the input amount and PCR cycle categories and the number of PCR duplicates across all of the 4 sequencers. The conversion kits generate more PCR duplicates, and of course, they also introduce, perhaps in the process, you introduce some contamination, but you definitely have several additional cleanup steps that might help you in removing the previously unnoticed adaptive primer dimers. The input amount determines the number of detected genes, but above 125 nanograms, it seems that there isn't much of a difference in the number of genes detected. But between 10 and 62 nanograms, there is definitely some variation, and it is caused by also the number of PCR duplicates, the PCR cycles and the PCR duplicates. Using the upper limit of the number of PCR cycles can generate data with higher proportion of PCR duplicates, as I already said, but UMIs, the use of UMIs is useful and recommended for removal of PCR duplicates, especially if you're using low input amounts. And I want to thank everyone who has been involved in the study, and they can definitely answer also all of your questions. Thank you.
Questions?
I'll ask.
Yes.
Do you plan to expand the study to include maybe other life reconstruction kits, and do you plan to maybe repeat some of the sequencing with freestyle?
Do you want to answer that?
Yes.
Yes, the first step is that we want to increase the number of samples. Yeah.
Yeah, we ordered the kits already.
Just a comment. There.
Go to the mic.
There are. I think it's fine. There are also some simple take-home messages from this. I'm very happy. We did this exercise. Is that you should always. I know it sounds very simple, but you should always aim for libraries within the range of the kit. We saw when you're lower than the kit, you will not get the. I know it's very simple, but this is a very useful resource, and the paper, when it comes out, we can always point to our clients, you have this RNA low input, because it will not work really well, even if you had experience before. The second is, we get a lot of questions from our clients about, you're using an Illumina sequencer, how will the data look like, will I see the same genes, different genes, other things. Very easy to point to a paper. The conversion step, does it affect it? Will it work if you're using within the library? We have something to point to. And the UMIs in the libraries, not many companies use them in their assays, but it really helped in the analysis. You can clean up your data. UMIs, they're really useful. So very simple, practical things, but having a resource to point to is a different thing than me saying so in the lab. Yeah, just wanted to say it was a great, thank you guys for doing this.
Sorry if I missed this. What was your threshold for calling detection? And then did you play with different levels, and how did that impact the overlap?
Gene detection.
Yeah.
Yeah, it was above 5 reads.
Okay.
Yeah.
And can you tell us anything about, this is a little rude, but can you tell us anything about duplication? Catherine introduced 3 other types on non-Illumina platforms. So do the other platforms suffer from optical duplicates, sister strand issues? Can you speak on that at all?
We haven't analyzed this across in the data.
Look at that
Yep.
yet.
Okay.
Yeah.
There's a lot of stuff that still needs to be done, so we want to get this thing out, so I think we have a good resource that we can follow. And hopefully, this is what to be expected, that you'll prompt you questions which will direct where do we want to take the study next couple of years. But it's just like the AVITI, it's just looking at PCR, right? But there are so many aspects of it.
Derek?
So, quick question on the AVITI, was the conversion done with a rapid conversion kit which uses PCR, or was it done with the standard conversion which does not use PCR and just uses ligation? Because that could be a source of PCR.
What's the answer?
duplicate.
That was the first one which was available so long before the rapid PCR. The very first version of conversion kit, just ligation, yes,
It was just the ligation?
just ligation,
Okay.
no PCR involved.
Don?
So the dynamic range of expression is quite wide, I think, in the liver. Did you see any effect within one condition of duplication of high abundance transcripts versus the low abundance transcripts? And did that vary from platform to platform?
So do I understand your question correctly? You mean...
Clearly, the amount of input has an
amount-
effect.
Yes.
But each input has its own dynamic range of genes that are expressed at very high amounts and very low amounts.
Aha, yes. Okay, no, we didn't look into that.
But you're using UMIs, so the amount of expression you get shouldn't affect.
You hope so, yeah, that's what I'm
if-
asking, yeah.
If the UMI space, for small RNAs, most of the kits out there, I'm sorry to say this, they don't have the UMI space and the complexity you need. But for gene expression using RNA-seq, the length of your UMI is usually more than adequate because your fragments are broken in random places. So even a gene, even if you have 10,000 counts or 20,000 counts,
Shouldn't be a confounding factor,
you
yeah.
have enough UMI space
Yeah,
to capture that.
Uh-huh.
But if it's a microRNA, for instance, and you have 50,000 copies, your UMIs do not often have the complexity
Right.
to capture that.
Right.
But so I think UMIs are enough for bulk RNA-seq, but if we were to repeat this for an amplicon-based assay or for a small RNA-seq assay, then I think breaking it based on the expression makes a lot of sense.
But we also see that the PCR duplication does create some bias, right? They're not, within the input amount, we did have, if we amplify, we do lose some genes. So the PCR duplicates are not created across the genes equally. But because we don't really have the biological knowledge of liver transcriptome, we didn't look at the transcripts according to what we expect.
You didn't see, you said GC content didn't seem to matter and
No.
things like that, yeah.
No, the genes that were, but we compared the GC content of reads detected between the different platforms, sequencing platforms, and no,
So,
there was no bias.
Another interesting
Yeah, and
addendum.
also to that, you also for the Illumina sequencers, your sequencing space is compressed due to the dimers you have. Because you have a lot of adapter dimers, your sequencing space is eaten up by that. So you don't have the ability to assess how it would look like without the dimers.
Sure,
What would be the duplication?
because there's finite
and
capacity
spectrum
on the
of
flow
that.
cell, yeah, sure.
So that's
yeah.
another
yeah.
layer of
So
complexity.
if you pre-amplify, linear amplification, because you're limited to a small sample, right, and then you have to go through some sort of linear, non-specific amplification just to bulk up enough, would that affect, do you think? Because now it's synthetic template instead of natural cDNA?
I am not sure. Does anybody have any experience with that?
I don't know. I don't know.
Interesting question.
It would go towards
Would you-
single-cell RNA profiling, as
would
it's
you
going to
be
be a
able
necessary
to
pre-step, right?
contribute?
Sorry, and then, so how extensible is this going to be to DNA copy number and even DNA variant detection for VAFs that are at low abundance in a tumor sample?
We should do DNA, but RNA-seq is a bit different, you know.
Of course, yeah, that's what I'm saying. So the stoichiometries here should be a little bit different, but the principle may be the same.
This exercise that we have this network going with just pure DNA parameters.
Sure, great.
We might repeat it on DNA.
Should be WGS or exome, that's another,
Of course,
maybe
yeah,
WGS, PCR-free.
yeah.
You can
Yeah, sure.
rejoin GRG.
Okay, sign me up.
Bring your sequencers.
Maybe I got a dumb question. Was this, were you doing paired end sequencing or not?
So some of the sequencing on some of the platforms was done paired end, but the NovaSeq 6000 was done single end, and so we only took the single end reads, the forward reads
I
for
see.
the analysis.
Yeah, because I mean, at least our impression, if you use paired end sequencing, you don't really get much difference from using the UMIs. It doesn't really add anything.
Okay.
Yeah,
Yeah.
because you have two fragments to
Right.
pick up for duplication.
Right. So you're thinking mainly for single end. It's
Yeah, so this would be a single end application, yeah.
Any more questions? All good.
Perfect, thank you.
So seriously, if you think you have ideas for where the study could go and what questions eventually be answered, send an email. We would love for you to join the GRG and actually participate in answering those questions, but at the very least, it'd be good to know that this is needed.
Yeah, plus these are done by dwarves, not the vendors themselves, so it's a, it's a different ballgame, so come on board, it's fun. Congratulations.
Thank you. Very good. Thank you for thank you.
We have some RNA-seq data if you guys want any of it to go into subsequent publications. I mean,
What kind of RNA?
do model our experiments.
Oh what RNA?
We just need to HR, big cycles, through a bunch of stuff. I mean, if you guys want.
Yeah.